So happy to see you guys here and to see many friends. And I've been thinking about these ideas for a number of months now, and I've been wanting to have an opportunity to be able to present them. So I'm going to go ahead and share my screen. This presentation, Maxwell's Angel, based on the thought experiment by James Clerk Maxwell from the late 19th century. And it's going to be about machines and thermodynamics. So let me start by reading to you a C.S. Lewis quote. He says, "The divine art of miracles is not an art of suspending the pattern to which events conform, but of deeding new events into that pattern. It does not violate the laws proviso." This is the natural law, or the laws of physics, we would say. It's not that law where it says A, then B. What it says, but this time, instead of A, A2, and nature, speaking through all of her laws, replies, then B2, and naturalizes the immigrant as she well knows how. She is an accomplished hostess. And so, I'm going to be talking a lot about the laws of physics, thermodynamics, and what we've learned. But I think the key to one of the things I'm going to talk about is how important information is in being able to create a machine that is able to do useful work. And that information to create the machine is these places where we as inventors or as designer, God has intervened in the past to be able to give a new situation. And so that's that A2, which leads to the result B2. And so that intervention is what's able to lead to miracles and efficient machines. So let's go ahead and dig into that a little bit more. So here's an outline for the talk. I'm going to start off by talking about steam engines and thermodynamics in general, just lay a little bit of groundwork, and then specifically talk about Maxwell's Demon, which is his thought experiment of thermodynamics and the second law of thermodynamics, and how information is very important for being able to create machines. So this is a little cartoon of Maxwell's Demon. Then I'm going to talk about specifically about efficient molecular machines, which we usually find in biology, but I'm also going to show you what we do as inventors in the chemical lab. And then lastly, because we're going to be talking about biological machines and all the information that's required to create them, we're going to talk about evolution and what evolution can do, what evolution can't do in terms of information input. Okay, so yeah, locomotives are just incredibly awesome machines. They're able to pull very heavy weights very far and actually quite efficiently too. And this is an example of a steam locomotive. And one of the things I want to point out here is that there is steam billowing out from all sorts of places here. And oftentimes people think that right up here in this central smokestack is actually where the exhaust is coming from that's doing all the work. But in actuality, it's down here at the bottom of the engine, right here on these pistons on the side that are actually doing the work and pushing the train forward. And you can see this clean steam coming out. This is actually the exhaust from the boiler mixed with some steam, but this has a lot of soot in it also. And so steam engines were being refined during the 19th century. This is the 1800s. And there's a lot of great scientists that were working to develop these. So let's look inside a little bit more and see how they work. So here is a cutaway of a steam engine. And you can see on the left, there's this large furnace. That's where the coal is being burned. And so it's very hot here. And the hot gases get pushed through these tubes, which are running through the boiling tank. And so you can kind of see it's a little bit darker here because they colorized it to show where there'd be water. And so running these hot tubes through the water, then and you get the steam, And the steam gets funneled down to the piston, which is right here. And this piston is then hooked up through the transmission to the wheels and pushes the engine forward. And so as we look at how a steam engine works, this is a little video that's a simplified view. Are you guys seeing the animation okay? So you're all supposed to be muted. I'll assume so. So what you have here is all the steam comes in from the boiler. So most of that engine that we saw was just a boiler to produce high pressure steam. And that steam comes in, and there's a valve right here that either pushes the steam to the right or pushes the steam to the left, depending upon the position of this piston. And so when the piston to the right, you want the steam to come in on the right where it's hot, it's under high pressure, and so it pushes the piston to the left. And while the piston is going to the left, the cold steam is being pushed out here through the exhaust. And then when the piston finishes its stroke, the valve switches and pushes the hot steam on into the left side so that you can then push the piston to the right. And notice also that as the piston moves back and forth, that there's actually this mechanism right here which causes the wheels to always rotate clockwise, no matter whether the is moving to the right or to the left. And so what happens is this offset cam actually will rectify the motion of the piston to always produce useful work in the forward direction. Okay, so locomotive efficiency actually has improved quite a bit. Back when an engine was first built by Newcomen in the early 18th century, the efficiency was down near 0.5%. And inventor named Smeaton was able to improve that to 1% over many decades. And then watt, and we have a unit of power called the watt. And so that's named after this scientist/inventor. He was able to improve the efficiency to 2%. And then we go a whole nother century until we're up to about a 7% efficiency. And then more recently, there's a whole firm that makes engines for large marine vessels, and they're hitting 54% efficiency. And so we are almost near the highest efficiency possible for an engine that just creates heat and then tries to use the heat to be able to turn it into to useful work. And so what I'm trying to show here is that with all these years of all the creative input of inventors using the scientific know-how of the scientists who study the phenomena of being able to convert energy, one form of energy to another, we've been able to drastically improve the efficiency of these machines. So this efficiency is actually a measure of how much inventive work has gone into making these machines. And so there's been some great scientists that have gone before us. For example, Carnot or Clausius, Boltzmann, and Maxwell. And you could tell from these photographs that these are pretty old scientists. All of them are dead now. And so thermodynamics had its heyday back in the 19th century. And we learned a lot about how things work. But one of the things I want to point out is that even though it's a very old science, this old science really needs to meet some of the new things that we've been discussing in the 20th century and now 21st century associated with information theory. They really didn't think about information back then when they were doing thermodynamics. And so let's combine information theory with thermodynamics and see what we can learn. Okay, so all of these engines, locomotives, are a type of heat engine. And so these heat engines, how they work is that you have some source of heat that is at a high temperature, and that heat flows into the engine, And that engine is able to convert some of that heat into work, and some of that heat gets wasted and goes around to its surroundings. And so we measure the efficiency by the ratio of the work to the heat that flows through the engine. And so these scientists studying thermodynamics, one of the fundamental laws of thermodynamics is the first law of thermodynamics, which states that the total energy of the system stays constant. But energy can be converted from one form to another. For example, we oftentimes use chemical energy in the form of fuel that has a bunch of energy stored in its chemical bonds. And then we do something to release that energy from the chemical bonds. In the case of a heat engine, you convert that energy into heat, and then that engine is able to take that heat and turn it into something like kinetic energy, which is able to push the engine forward. So kinetic energy is the energy of motion. Now, we can convert from one form to the other, but you never get more energy out than you started with. That would be a perpetual motion machine. And then we get to the second law, but let's first describe what is temperature. And so temperature is a measure of the average energy of all the particles. And so when you have very fast moving particles, this is a graph of the number of particles as a function of how fast they're moving. So this is the velocity. And you can see for a cold gas that's at 100 degrees Kelvin, there's this distribution where most of the molecules are moving below 500 meters per second. And there's some faster ones, but a lot of slow ones too. And then as we go to a higher temperature, let's say 1,000 Kelvin, then most of all the molecules are moving faster than these molecules that are moving at 100 degrees. But, some of them actually share the same speed, but those are the coldest ones. And so for each temperature, you will have an ensemble of molecules that some of them are going fast, some of them are going slow. But the temperature is a measure of the average energy of those particles. And so what the second law of thermodynamics says is that if you have an isolated system, that it will spontaneously evolve toward a state of maximum entropy. And what that looks like is here on the left are states of low entropy. One of them is where you have hot molecules on the left separated from cold molecules that are moving slowly on the right. And if you were to let this system sit, what will happen is there'll be heat transfer from these hot molecules as they tap into the cold molecules. They'll transfer their energy, and then it'll equalize so that on average, the molecules have all the same energy throughout. Now remember, there will be that distribution of energies depending upon your average temperature in the system, but it will be more homogeneous. Another example of the second law of dynamics is that, let's say you have a glass of ice water. Well, the ice is actually a very low entropy state. And so over time, what will happen is the energy that's in the warmer water will mix with the energy of the colder water, and it'll melt the ice so that frees those molecules to move around all over the place. And the freedom of those molecules to move all over is actually a higher entropy state. And so this system will evolve towards this system, which we see all the time in nature. Ice melts. Okay, I just want to describe a little bit more accurately what is entropy. And so I simplified it to, let's say we had 4 molecules, and those 4 molecules can occupy a system that has 16 spaces available. Well, if we were to keep these 4 molecules right next to each other, such as they would be if they were frozen in ice, there would actually only be 9 configurations where all 4 of those molecules are right next to each other. Now, if we were to let those molecules go all over the place, free from each other, where they don't have to be right next to each other, and we started counting up all those configurations, we'd actually find that there's 1,811 configurations where the molecules are not all right next to each other. So there's a whole lot more configurations where the molecules are separated from each other. In a sense, I'm saying that it's much more probable that it exists in a more random state than it would be in one where they are right next to each other. And so this probability of it being in a more random state is actually just a restatement of the second law, that it's more probable. And I have this equation down here. This is actually the Boltzmann equation. Remember, one of those famous scientists was Maxwell Boltzmann, and he came up with this explanation or derivation of entropy, where he said that entropy is just a measure of the number of states or configurations that you have and the log of that. And so we would actually say that the entropy of a system with 4 things in the 16 spaces would actually be Boltzmann constant times the log of this plus this, which would be around 3. Okay. Any questions yet? I'll pause for you guys to be able to unmute and ask any questions about what is entropy or the second law of thermodynamics. Okay, if not, let's jump into the next section, which is going to be discussing Maxwell's Demon and information and how you might make a machine. So remember, Maxwell was one of the scientists that I gave the picture of on the right. Maxwell was the son of a Presbyterian minister and actually a devout believer himself. And he was thinking about this second law of thermodynamics, that systems that start in a low entropy state would actually move to a high entropy state. But he was thinking, well, if we were to separate these two gases, which are very similar, actually they're the same in their temperature profile, and we were to have like a little door in between the two sides, then we would have a demon that could open and close the door. And the demon would say, wow, a hot molecule is coming from the right side. I'm going to open the door and let the hot molecule through. But whenever a cold molecule is coming through, the demon would close the door. And that way the molecule bounces off. So we'd hold the cold molecules on the right side, allow the hot molecules on the left side. And over time, what would happen is you'd get to a system that had all the hot molecules on the left, all the cold molecules on the right. In a sense, this would be a violation of the second law of thermodynamics. Is this kind of thing possible? It'd be like reversing time and going from a glass of water that is even in temperature to one that has ice on one side and warm water on the other side. Or we saw the piston on our steam engine before. The equilibrium state would be where the gas is the same temperature on both sides. That would mean it would also be the same pressure, and therefore it would push the piston. So the piston would just stay there. But instead, on an engine, we see that we put the hot gas on one side, that's at higher pressure, and it's able to push the piston to the other side. If this demon were to be able to see these molecules and open this door at just the right time and get all the hot molecules on one side and all the cold molecules on the other side, we would be able to freeze ice or make a perpetual motion machine, which pushes the piston to the right. Now, it actually doesn't need to be a perpetual motion machine. Can we configure things to be able to make cold on one side, hot on the other side, without being a perpetual motion machine, but just a machine, to be able to get things like ice and a piston that's able to work? And the answer is actually yes. We see it all the time. You go to the refrigerator or the freezer, you can pull out an ice cube from the freezer, make yourself a nice glass of ice cold water. Before the invention of refrigerators, you could never do that. Or you could go out to your car. Most of us have internal combustion engines in our cars, and you start that engine up, and what's happening is you're getting hot gas on one side of the piston, and it pushes that piston down, does useful work, and is able to drive the car. We are used to seeing this all the time. So are our machines, our refrigerators, and our internal combustion engines a violation of the second law of thermodynamics? Because we're able to get hot gas on one side, cold gas on another? Well, let's look into Maxwell's Demon a little bit more closely. So the first thing that we notice as we study Maxwell's Demon is that if we have a door that is opening and closing, let me first say, if we have a door that is able to allow a molecule through at one time and deflect that molecule at another time, that door actually has to have some substance to it. It needs to be able to apply force to that molecule. And it could either be because it's massive, or because you could create like an electric field barrier that can repel the molecule. But either way, the ability to open and close that door requires you to put energy into the system to either move the door if it's massive, or charge up a field if you are using some type of electrostatic deflector for the molecule. And so to be able to effectively function, Maxwell's Demon requires energy. Can't get anything for free. And as a matter of fact, when the demon is working and he's applying his energy to the door to be able to open and close it, what is found is that he actually puts in more energy than what is created by the separation of the hot molecules from the cold molecules, and the whole system warms up, which is actually another statement of the second law of thermodynamics, that the energy of the universe always increases, or the entropy of the universe always increases whenever you do something. And so this poor demon, he's very sad. He wasn't able to beat the second law of thermodynamics. But another thing that we see is that Maxwell's Demon doesn't only require energy, but it also requires information. The demon needs to know when to open the door, like right now when the hot molecule is coming, and when to close the door. And that sequence of being open and closed, closed, open, closed, open, closed, open, open, that sequence of states is a specification. If I were to just open and close the door randomly, then sometimes cold molecules would go through, sometimes hot molecules would go through, sometimes hot molecules would bounce off, sometimes cold molecules bounce off. There would be no productive work or no separation of the hot molecules and cold molecules from which to do useful work when you have the hot system. And so this information is extremely important for having a functional demon to be able to separate those hot molecules from cold molecules. And, oftentimes we're used to seeing information in ones and zeros. Actually, maybe not very many of us work in binary day to day. But let me tell you about some other information. What about if I were to say, what you should do is you should fashion a piston that fits tightly in a cylinder. And then you should fashion an opening at the ends of the cylinder and fashion a boiler to create high pressure steam. You should fashion also valves that direct the hot steam into one side of the cylinder and release the pressure at the other side. And then that valve switches when the piston has finished their stroke. And then you should connect a load to that piston and have that load be rectified, have that load to the piston be rectified for the reciprocating motion. What I mean by that is, it's something that reverses the motion so that the reverse motion of the piston doesn't cancel work already performed, but further extracts more work. So what I've basically just done is I've described that steam engine we saw a few slides ago. Because if we can fashion a system to be able to get a hot gas on one side we can push that piston over and produce useful work. And, this isn't actually much information. If you were to just go and take what I've written here, you might be able to get an engine as efficient as those first steam engines that were like 0.5% efficient. I'd have to give you a whole lot more information, like give you some very tight tolerances on how big the cylinder should be relative to the piston so that the piston can actually fit snugly but still be able to slide. In machining, we measure things to fractions, less than a thousandth of an inch in diameter or inch in length to be able to get really precise fit of these pistons and cylinders. And I can go on and on and on, giving you more and more information about how to build a better steam engine to be able to get to the high efficiency. So it really requires information to be able to create a good machine. And as a matter of fact, we can think of all sorts of examples of where that information is lacking. For example, here's a bunch of fuel, and we can burn the fuel. And if there's just no machine there, you get absolutely no work. And so what this heat engine here on the left would look like is you have a big heat source and you got a cold sink, and you just take all that heat and you put it to the cold sink. No work done. Or what about if, you haven't had a mechanic continually putting in new information into your motor and things break and go wrong. You might be able to burn fuel in that motor, produce a lot of heat, but that car is not going anywhere. And there's a whole spectrum of, how much information is there. For example, on this car, its motor has a blown head gasket, which means that things aren't fitting so tightly. And when the fuel is burnt inside the cylinder, instead of that energy going into pushing the cylinder down, you have a lot of that energy slipping through and pushing water around and also pushing water into your cylinder. And so that's why you get so much steam coming out the back here. And so because not all that expansion of gas is being funneled into pushing that cylinder down, you have a reduction of work. But you could actually still drive this car. It might get 6 miles per gallon then, instead of the normal 20 or 30. So information is very essential for machines, and information can actually be lost. Okay, so as we think about machines, one of the ways to actually be able to optimize the efficiency is actually to work near reversibility. What I mean by reversibility is that the engine might actually go in the forward direction just as easily as it goes in the reverse direction. I should have reversed that. That the engine could go in the reverse direction just as easily as it could go in the forward direction. And in terms of heat engines, like steam trains or this diesel engine for a large ship, reversibility is achieved by going quite slowly. And so you can see that this is a big engine going at around 500 RPM. And by doing that, they're able to get about 54% efficiency for the fuel. Now, this is a really big engine. It's actually a 180 liter engine. This is actually like the height of 3 people tall. It's a really big engine. And the power density in this is about 11 kilowatts per liter. But we don't care as much about efficiency in our engines and our cars. We actually care a whole lot more about power. So we want something that's going to be able to give us like 100 kilowatts per liter. And so we do that with like a little 1.5. This is a Honda engine that would go in a Honda Civic these days. And they're about 30% efficient. But notice that they're working much faster at 6,000 RPMs to be able to achieve this power. Okay, now you can achieve reversibility by going slower, and that's what Carnot emphasized when he was talking about efficiency in his engines. But another way to be able to go to higher efficiency is actually to work at the much smaller scale, to work down at the nanoscale. So that brings me to the next section of my talk, talk about making efficient molecular machines. Anyone have any questions about Maxwell's Demon and, is it possible to violate the second law of thermodynamics? No, but definitely be able to get useful work out. We have to use both information and energy. Any questions there?
I have one question. I guess the Demon has to open the door to let the cold molecules go to the right, just as much as it opens the door to let the hot molecules go to the left. Is that right?
Yes, that could be part of the information content.
Otherwise, you'd never get rid of the cold ones on the left. There'd always be a mixture of hot and cold molecules.
Yes, that's true. And so, we would keep trying to work as much as possible right at these tails. And so, you could get separation of heat by only thinking about letting hot molecules to the left, but you would get better separation of heat by actually taking the cold molecules from the left and putting them to the right. So that would be a more effective machine. So in a sense, Mark, what you're saying is we need even more information to make an even better machine, right?
Yes. Thank you. Thanks for the clarification.
And Bob and then Patrick, do you have questions?
I'll let Bob go ahead.
Saying that there's a lot of times people have invented things and they forget about the larger, that you have to look at the whole system there. You can't just look at one isolated part there. This carries over. I'm thinking of, I think it's Major Barbara, for example, the play where you have this person manufacturing weapons, and in this one place, it's a wonderful standard of living. People are, busy, they're, food and so forth. But that efficiency, or what you call it, or that thing, is at the cost of the fact that there's elsewhere, they're spreading, entropy, devastation, blowing up things there. The same thing as a broken window fallacy there. I think one of the things that, at least in the social sphere and the life sphere, is that, thermodynamics teaches is that we have to look at the whole, we have to look at everything and not just the one little part that looks appealing there.
Mm-hmm. Mm-hmm.
And of course, you can't.
That's interesting to apply that.
Law. No, sorry. No, but anyway, but yeah.
That's interesting to apply it to social aspects to it, social sciences.
So I think the concept is.
I'll sort of second what Bob said, because as a trained systems engineer, one of the things that you do when you put the system block diagram together, you need to know where to draw the dashed lines for where you're going to define the system or subsystem within about which you're going to do the analysis. So that's a good concept to sort of keep in view. Paul, I think you answered with the pictorial and the two engines that were higher efficiency on the previous chart, what I was going to ask. And when I first saw the list of the increased efficiencies that you showed earlier, the question arose, and actually it was my wife that brought it up when she saw the chart, were the high efficiency engines necessarily steam engines? And the answer is pretty clearly no. And I think you're probably going to take us into a realm where these even higher efficiencies are far from the sort of fuel burning kind of locomotive engines that we're used to. So I'll be eager to see that. Just the other comment I was going to second Mark's notion, because I did a little refresh myself on the Maxwell Demon problem. And yes, the Demon does have to be able to open doors to let the hot ones into the hot chamber and let the cold ones into the cold chamber that aren't already in those respective chambers. So, and as you pointed out, Paul, that requires an increased bit of information. He has to be able to have eyes on each of the compartments and to be able to discern the velocities, the molecules that are approaching the little trapdoor.
Mm-hmm. And so you pointed out that these engines right here are not steam engines, and I would say though that whether it's an engine or a diesel engine or a steam engine, they all are of one category, which is called a heat engine, where you burn your fuel and you create heat, and then that heat does some work, and some of that energy is dissipated, and they all end up following the same thermodynamic principles, and they all end up having the same limit of efficiency, which is called the Carnot efficiency.
Right.
I used to have a slide in here, but I thought to explain Carnot efficiency might be a little technical, so I actually dropped that slide. But whether it's a steam engine or a diesel engine, they all end up following the same limits of efficiency. Now, or also gas turbine, like what we produce electricity from.
Right.
And so I think that's an interesting point, is that the efficiency depends upon the temperature differential between the heat source and the cold source.
Right.
And so these engines here tend to be run a lot colder because we are concerned about emissions. And if you run them too hot, then you produce a lot of nitrous oxides, which are really bad for the environment. We have a lot of difficulty scrubbing them from gasoline engines. We can do a little better on these marine ones, and these ones also burn out at sea, which we kind of don't care about emissions. But on our electricity production, we run the temperature really high, and then we are able to scrub off the nitric oxide. And that allows us to be much more efficient when producing electricity, even though electricity generation is still a heat engine.
Do you have just a quick off the top of your head, and I don't want to divert, it's the last thing I'm going to ask about. What the rough efficiency of, say, an automobile hybrid engine is, where you have a combination of, gas powered and also the battery? Any idea what those efficiencies run?
They're like at 40%.
Okay. Yeah.
Because like a Prius is just a Toyota Corolla with the hybrid, and so a Corolla gets like 35, and then 35 miles to the gallon, and then a Prius gets like 45, 50 miles to the gallon.
Okay, good. Thanks.
Yeah, but in doing that, you have to consider the fact that it takes a lot. You have the energy to put into the battery part. That's being expended somewhere else at the manufacturing plant there.
No, the energy that goes in the battery comes from the kinetic energy of the car, which got there from the gasoline.
Oh, you're saying, that one is not coming. You're not charging the battery externally.
Yeah, not charging the battery externally.
No, okay.
And so, while we're talking about batteries and efficiency of cars and stuff like that, I mentioned already that steam turbines, because they run so much hotter, are able to get much higher efficiencies, up near 70%. And if we have like 10% transmission losses through the high voltage lines to residential electricity, and then you have like 70-80% round cycle efficiency on charging a battery and dissipating a battery in an electric car, in principle, electric cars could be more efficient if all you care about is how much fuel goes into creating the kinetic energy. Now, granted, electric cars are very expensive. Okay. Thank you for the questions. Any others? Yeah?
Can you speak a little bit about what work is in this diagram? Is it always mechanical, like rotation? Or can you have other things like, chemical reaction is also considered work?
For these kinds of engines, it's always some type of kinetic energy. And so if it's linear kinetic energy, then it's just force times distance work. If it's rotational, then you have some torque and some rotational angle that you apply the torque over in terms of how much work is done. But these are all some form of kinetic energy that is the work being done.
Okay, actually, I had one more question.
Yeah.
Going back to your definition of entropy, where you had the two different configurations.
Yes.
how, my understanding is that, the number of configurations for some kind of a macro state, right? So you've defined a macro state of 4 being close to each other, whereas the other ones are 4 not close to each other. Is that correct? And so if the macro states are, let's say, more specific, then you would have different numbers of configurations, and each one of them will have different microstates?
Each one will have different microstates. Are you wanting to say, like, you want to differentiate having 2 molecules next to each other compared to
right? For example,
all of them apart?
Let's say the configuration that I have is all in a row. Then there would only be 4 configurations, right?
Okay. This is a little subtle, but
8 probably.
Even though I separated these 9 configurations from these 1,811, these are actually all the same set of states at the same entropy. And you might say, oh, Paul, you're contradicting yourself. But what I'm saying is even though these are included in the same set of states that make up this entropy, the 1,820 states that we take the log of multiplied by Boltzmann constant, because this is so much more probable in nature, you're not going to see these condensed states. That's why we see, gases spread out instead of it condensed on the corner when you have a system that is isolated. But we would actually include these 9 states amongst all the configurations when calculating the entropy of this system of 4 things taken within a space of 16 possible positions in space.
Mm-hmm. Okay. Yeah.
And so I know I'm not really answering your question about microstates, but I think that actually adds a little bit too much complication in how we do the counting.
Okay.
Okay. Charles is waiting to ask a question.
Okay.
Yeah. Maxwell's demon is active. I'm thinking about a passive selector, such as if you had square objects and triangular objects, and you put a triangular hole in the membrane, the triangular molecules would slip through, but the square ones wouldn't. So over time, if there were energy in the system, you would get triangular molecules on one side, well, more, and square molecules left on the other side. And that doesn't seem to require any work or information to me.
Now,
and I think that's how biological. Yeah.
The thing is that those triangles could slip back the other way just as easily.
Oh, that's true.
If you were not actively selecting. And as a matter of fact, let's say you started with all triangles on the right and all squares on the left, and you had a hole that could only allow the triangles through. Then what would happen is the triangles would become more dilute on one side, and they would pass over, and it would reach the highest entropy state, which would actually have more units on the side that has squares and triangles, and far fewer ones on the right side. And that's actually what we see with when we do osmosis. You can actually end up with when you have osmosis, you start with like a saltwater solution, and if you have a salt impermeable membrane, but that can allow water through, and then you put it next to water, what will happen is water will pass through the membrane to get to the salt to help dilute the salt, and you'll actually end up with a higher pressure because of all that water that's moving through. And so you might say, well, higher pressure is not an equilibrium thing, but that's actually because the entropy is driving the water over to reach a lower free energy state.
Okay, that's helpful.
And we're going to talk about nanoscale molecular machines, and there's a lot of passive channels in biology. What I mean by a passive channel is that channel where you have a membrane, and that channel could allow molecules through, and it's selective to only certain kinds of molecules. Some channels are passive, meaning there's no way for the channel to use another molecule as fuel to actively transport. And some channels are active, meaning they can actually use a fuel molecule to, in combination, to push the ions or the molecules through. When you have a passive channel, all they do is they just allow enough molecules through to reach equilibrium. And so that they allow you to get to the point where you have the same amount on both sides, or the same concentration on both sides of the membrane. Usually those passive channels are paired with active channels somewhere else, where the active channel is like using work to push and push the system away from equilibrium.
Well, there's also the process of creating that barrier in the first place. It doesn't just happen out of thin air. You have to create a barrier that has a selective permeation.
Right.
So that's either imposing information, or you're imposing some kind of work to create that barrier in the first place. So it's not a free lunch there.
Yeah, definitely. It requires information to be able to create that barrier. That's a good point, Bob.
Okay, thank you.
All right. Any other questions? Okay, let's start talking about efficient molecular machines. So I commented how on the previous slide that reversibility is one way of achieving high efficiency. And at the macroscopic scale, we do that by going very slowly. But you could actually go fast and be reversible when you go down to the nanoscale. And so I want to start by pointing out that people are interested in making nanoscale machines. We even gave a Nobel Prize in 2016 for making molecular machines. And some of the kinds of molecular machines that have been made is, you can see this molecule here. And what happens is there's this ring that goes around this chain of atoms, and the ring cannot come off either side because it's really bulky here on the side. So the ring can't slip off. And so the ring is stuck in between here. And depending upon if you add an electron to the system and charge this one up, you will either have the ring occupy the left side, or if you take that electron back off, then the ring will be on the right side. And so by changing the chemical potential, or the redox chemical potential, to be able to add electrons or pull them away from this molecule, you can actually move this molecule back and forth and do useful work. Now, this is not the state of the art. This is actually from the Nobel Prize literature, that they said these inventors did this, and you know how Nobel Prizes are oftentimes very delayed from the actual invention. So this work was actually done in the 1990s, but we haven't actually gone that much further. So, for example, this is what was done in the mid-2000s, where we can put these round molecules at the end of these chains of something that's kind of rigid. And so these molecules sit on a very flat metal surface, and these have no way of processing any fuel or anything. But if you come along and you kick this thing with a scanning tunneling microscope tip, then you could actually give it some energy, and then it will cruise around on the surface a little bit, and then it'll stop after it dissipates the energy. Then you could come and you could kick it again. But this tends to go in a certain direction. Why? Because of the positions of these balls are just right to be able to line up with the lattice of the metal atoms here, such that it'll go along with that lattice, and you could have more useful work in one direction instead of it just kind of being random. And so this is what we do as humans. These are our molecular machines. And let me tell you, these things are not efficient at all. We are back kind of like with the Newcomen 0.5% efficient, and we're actually less than 0.5% efficiency. But let's see what exists in nature. And so this video is so good that I am going to just let us listen to this video. This was put out by the Stewart Lab at Harvard. And this is not Stewart in the Chemical Sciences division, where I said Chemical Science division. I mean the Department of Chemistry and Chemical Biology. But it's over in the med school.
Life requires energy. The universal biological fuel is a molecule called ATP, or adenosine triphosphate. ATP stores chemical energy in the form of a high-energy phosphate bond. As the name suggests, it consists of an adenosine ring with a chain of 3 phosphate groups attached. The last of these phosphates can be split off to release the energy within the bond, resulting in adenosine diphosphate and free inorganic phosphate. This energy is needed to keep cells alive and is used from processes from muscle contraction to producing the thoughts your brain is having right now. The hydrolysis of ATP can be reversed by the addition of inorganic phosphate to ADP. However, this of course requires energy and is performed by a molecular power generator called Atp Synthase. Here we see an atomic model of F1-FO ATP synthase sitting in a membrane. This molecular motor is found in the mitochondria of eukaryotic cells and is responsible for making most of the ATP. Mitochondria, the powerhouses of the cells, turn over around 60 kilograms of ATP every day in an adult person. A long chain of biochemical processes convert the energy stored in food into a proton gradient across the inner mitochondrial membrane. These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of 2 separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin, and at the top we have the F1 motor from factor 1. The FO motor is a proton-powered motor. It is thought that protons flow through a channel open to just the intermembrane space, where they bind to a ring of protein subunits, rotate 360 degrees, and exit through another channel exposed only to the matrix. The net flow of protons driven by the proton motor force provides the energy for the generation of rotation. The torque generated in the FO motor is transferred to the F1 motor by a central stalk or shaft. The F1 motor is responsible for generating the ATP by addition of phosphate to ADP. The top of the central stalk acts similarly to a camshaft, so that as it rotates within the F1 motor, it causes conformational changes of the catalytic subunits. The catalytic unit is made of a dimer of subunits, and there are 3 of these arranged in a ring. Catalysis occurs at the interface between the dimers. If we concentrate on one dimer, we can observe 3 distinct states. First, ADP and phosphate bind to the catalytic site. The central shaft then rotates 120 degrees to rearrange the molecules. Next, the enzyme undergoes a further 120-degree rotation, and the ADP and phosphate are fused together to create ATP. The enzyme then rotates again to return to the starting position, where ADP is released and ADP and phosphate can be bound for the next cycle of catalysis. One key aspect of this relationship is that catalytic subunits must remain stationary with respect to a rotating central shaft. This task is performed by a scaffold on the outside of the complex, referred to as the peripheral stalk. Recent work has shown that what was once thought of as a rigid scaffold is actually dynamic and is able to accommodate the changes necessary for most efficient function. Looking at this machine in its natural environment by electron chromatography of intact mitochondria, work has shown that ATP synthase dimerizes to shape the mitochondrial inner membrane into their signature cristae shape. This turbocharges ATP synthesis by focusing the proton gradient near ATP synthase.
Okay, so what you have is, here's the membrane of the mitochondria, of the cristae, which is within the mitochondria. I'll explain what mitochondria are later. But there's more protons, which are hydrogen atoms, on the bottom side than there are on the top side. And as we were talking before about these pores that are able to actively transport one type of ion or molecule from one side of the membrane to the other, this is one of those active transporters. And so I was emphasizing before that there's reversibility, meaning it can go forward as easily as it could go backwards. And so if you have a lot of ATP, this thing's used to create ATP, but if you have a lot of ATP that's already been created, this thing will actually work backwards, and you can use it to pump protons from this side to the other side. Now, it's usually used where there's more protons or hydrogen atoms on this side, in which case then it moves, I'll call that forward, where they move around by first coming through here, binding to this stalk, and then the stalk rotates around, and on its way around, then the protons are able to leave and go to the other side through a different channel. And so the specific configuration of this channel allows the stalk to rotate as it's wanting to allow the protons through to the other side. And the potential from the higher concentration to the other side is what drives that. Now, it's going to be rotating the stalk. And what this stalk does is it rotates within these F1 fragments that are up here. There's 6 F1 fragments where they are organized into 3 pairs. And in the interface between each pair is where the ADP and the phosphate bind. And as the stalk rotates, it causes those to move relative to each other, and it traps and actually puts the mechanical energy into forming that bond, that high energy bond, to create ATP. And so as it rotates around, it first binds them, then it pushes them together, forms the bond, and then opens up and releases, each with a 120 degree, each of those steps requires a 120 degree rotation of the stalk. Just a absolutely phenomenal motor. They're about 100 nanometers in size. You can't see them with a regular optical microscope. You need a good electron microscope to be able to see them. And they're incredibly efficient too, because they're completely reversible. Okay, so let's talk a little bit more about these kinds of machines. And so you guys might be familiar with the energy cycle, where we get energy from the sun. Plants use light to be able to do photosynthesis, which creates food and oxygen. And we consume the food and oxygen, and the oxygen gets into our blood and somehow gets formed into sugars. That's a whole nother set of biochemistry lessons, because we normally consume our food in some form that looks like this. As a matter of fact, I very much like to consume this right now. And let's say you have sugar and oxygen. Well, then we burn that sugar and oxygen to create energy, water, and carbon dioxide. And that carbon dioxide goes back out. We breathe it out, and it goes to the plants, and they use that light and carbon dioxide to make our food and oxygen. So, there's this constant cycle. But let's just look at it from the point of, if you have sugar, what happens? And so we could take sugar and we can heat it up and burn it, and we could get energy out of it. But this would actually be an incredibly inefficient process. This would actually be the same kinds of process that we were talking about with the thermal engines before, where we could at most be the Carnot efficiency, typically 30%, 50%. And actually, because sugar is a solid, it's a really bad fuel. And so it's hard to make a good engine out of it. It gets gummed up. But, when we take those same sugars and we use them within our biochemical machines, we get a lot more energy out of it because it's done so much more efficiently with reversible molecular machines. So what do some of those look like? Well, first, let's just start with glucose. And the way to be efficient is to make very, very small steps in the energy as you burn it. And so instead of taking all the atoms and blowing them apart like you do when you make a flame, instead we're going to move one atom at a time or a few atoms at a time and extract the energy at each one of those. And so glucose to the glucose 6-phosphate. And, how do you make this transformation? Well, there's a molecular machine for that. Well, once you have the glucose 6-phosphate, and you need to go to the next step, well, there's a molecular machine for that too. Actually, there's a molecular machine for every single one of these steps over and over again. There are dedicated molecular machines for doing each one of these steps efficiently and reversibly. And actually, sometimes you give up on the reversibility, but these are designed to be quite efficient, even if they're not totally reversible. And so this takes us from glucose to this molecule that has now split the 6-carbon ring up into 2 molecules that have 3 carbons each. And we're using oxygen and water all the time for these. And on many of these steps, we are pulling off some of these high energy molecules like ATP or NADPH that are used later on to help fuel events in the cell. Well, that pyruvate then gets put into what's called the citric acid cycle. And again, a lot of chemical modifications are made to make these molecules. And look, there's molecular machines for every single one of these little steps along the way. And this citric acid cycle is what's done inside the mitochondria. The mitochondria is one of the organelles inside a cell. And so this cell is about 10 microns in diameter. Remember that a millimeter is like, 1 millimeter is 1/1000th of a meter. And so a millimeter is like the width of this screwdriver that I'm holding, the shaft of the screwdriver, okay? Most screwdrivers are bigger and stronger than this. They're maybe 4 or 5 millimeters. This one's a pretty small one. It's 1 millimeter. And if you take that millimeter and then you divide that into 1,000 equal steps, that's called a micron or a micrometer. And to be able to see things that are about a micron in size, you need a very good light microscope. And individual cells are about 10 microns in size. A bacterium, which is also a cell, those are about 1 micron in size. But these larger, more complex eukaryotic cells, which make up our body, they're about 10 microns in size. There can be a lot of variation, but they're at least 10 microns in size. And inside, there's a bunch of these mitochondria. Now, these mitochondria are often about bacteria. So from this end to this end, it's about 1 micron. That's 1/1,000,000th of a meter. And if you take a micron and you split that up into 1,000 individual units, those are called nanometers. And atoms and molecules are at the size scale of nanometers. And this mitochondria is about 500 nanometers along its short axis here. And you can see these lines here. Those are membranes. Those are those inner membranes right here. And there's a bunch of molecular machines that are used for pumping the protons from the inside of this matrix to the outside. And then those protons come back across through the ATP synthase to make ATP. And so this molecule right here, this ATP synthase, is the one that we just saw the video on. But these other ones are used to be able to create that proton gradient from one side to the next, which drives the ATP synthase. And each one of these are also these amazing molecular machines. And this one, complex 3 right here, is actually this succinic dehydrogenase that is part of this citric acid cycle. And so let's look at that. So complex 3, I'm sorry, I said it wrong. Complex 2, right there, the cytochrome C, is the succinic dehydrogenase. And these, this chemical process to dehydrogenate the succinate actually drops some electrons on these molecules right here within this protein. And then those electrons are transported down until they get to the ubiquinol to make ubiquinol. And then that ubiquinol is passed to complex 3 right here. And you can see that it comes and binds to this cleft. It has its electron, it passes those to these other ones, and then those electrons move around and are able to do the work of transporting these protons across the membrane from one side to the other so that ATP synthase can work. And if these molecules are not positioned just right, then these electrons don't conduct, they don't do the work, you lose efficiency. But these are precise, these are held in just the right position to be able to be very efficient. And these are just a couple of the molecules that are part of this citric acid cycle or this glycolysis. So many very precisely organized molecular machines are required to do these steps to burn our fuel efficiently. And what are these proteins? Well, these proteins are actually these sequences of amino acids that are folded up into these precise structures. And the amino acid sequence actually reads just like letters in a sentence in a book. And the organization of letters gives it its its shape, which gives it its function. And so just like there's certain organization of letters that produce meaningful words and sentences in the English language, so also there are meaningful sequences of amino acids which give functional proteins, while most of them actually do not produce functional proteins. And that was what we were talking about last week with the Douglas Axe book. And this sequence of amino acids is actually, that information that's stored here is actually stored in DNA. And so it's kind of striking that this sequence information is very much like that sequence of ones and zeros that we needed to be able to get the Maxwell's Demon thought experiment to work right. Information can be reduced to sequences of ones and zeros. Okay, so we talked a lot about biochemistry just now, and I presented a lot of it. Normally, you don't see people present a lot because I wanted to kind of give you that wow factor of it's just so amazing that all these things need to work together. Any questions on the biochemistry? Did I lose anyone there?
I have a question about the reversibility.
Yes.
Are these really perfectly reversible reactions? I thought that in order to be reversible, they had to move infinitely slowly.
Okay, reversible means you can go backwards or forwards, and it does it on average the same. And when you're talking about big things, reversibility is slow for us, but as you go to the nanoscale, things move a lot faster. And let me try it this way. Why you need to move slowly on big machines is because you actually want the energy to be distributed evenly throughout the whole machine all the time. And so when you give an increment of energy, you need to wait for that increment to spread out through the whole thing. And that way it could move forward as much as it could move back. So another way to state reversibility is that you could take the energy out as easily as you could put it in. When you move to a very small thing, the time that it takes for that energy to diffuse across the whole machine is much smaller because it's so much smaller. And so the system works where the energy is evenly distributed much more easily at the nanoscale. And so you can put energy in and you can extract that energy out just as easily. And I'm going to throw a little bit of technical work at you. It has to do with the dissipation fluctuation theorems. And so it's a matter of how easily do you dissipate energy. And at the nanoscale, you dissipate energy as easily as you pull energy back out. Another way, I'll just keep throwing a few technical terms, is at the nanoscale, we're working at very low Reynolds number. And Reynolds number is the measure of the inertia of something to its dissipation. And so at the nanoscale, the dissipation is much higher compared to its inertia, and therefore you can dissipate energy or you can pull it back out of the system.
Thank you. Does that mean that they're nearly perfectly reversible, but not quite?
The ATP synthase is completely reversible. And are complex 2 and complex 3 reversible? Yes, they are. These are completely reversible right here, these 2 examples. On the previous slide, these are all completely reversible. And some of these are not reversible. This one definitely is, but this one isn't right here.
Well, final question along these lines, though. If something is completely reversible, what determines which direction the reaction goes?
Okay, it's going forward and backwards all the time. It's just on average, we would call the direction that we consider to be metabolism to be the forward direction. And the reason why is simply from concentration gradients. And so when we eat, we end up with more fuel. And so when you have more of the fuel molecules available, when you have more of the glucose, then it pushes this reaction to the right. That's Le Chatelier's principle from general chemistry. And what happens then is you have some of this. Well, if we're depleted on the pyruvate, then they'll push this reaction this way. And that's going to push this reaction this way, and this reaction this way, and this reaction this way. If for some reason we end up with a lot of pyruvate, then the reaction would actually start going back in the other direction, and we could make glucose. Okay. Well, this one's a little irreversible, but it's just a matter of balancing concentrations. And when you end up with more concentration of one, it pushes the whole ensemble of reactions in what we call the forward direction, which is metabolism. And so why is that the forward direction? Because what's happening is ATP is being taken, it is diffusing around the cell and is being used by other molecules to do its productive work. And so as the cell moves cargo from one side to the other, or uses ATP to make some other concentration gradient that is useful for the cell, then ATP is constantly being depleted, which then means that the protons are going to want to go through ATP synthase. And if we have very few protons here, then we're going to want to be able to have the electron transport chain functioning to be able to transport protons across. And that means that you're going to need the succinate binding here. And well, to get to the succinate, you need the citric acid cycle to move forward and stuff like that. Does that explain it?
Thank you. Yes. Thank you.
So Paul, can I ask a follow-on, sort of a dumb question? Because I'm not anything close to a microbiologist, but if I understood the dialogue that you and Mark just had, if this process of, I inferred that you said that metabolism is a reversible process. Is that fair to say or not?
These steps in metabolism, most all of them are reversible, yes.
So what do we call when it goes the reverse direction? I mean, it's not demetabolizing, but I mean, what is it? What's happening?
It's at equilibrium is what's happening. At equilibrium, it's not that the reaction stops happening. Equilibrium is when the reaction forward equals the reaction going backwards. Chemistry is happening all the time. And so you have some of them that are going forward, some of them that are going back, but on average, you don't change concentration. That's what we call equilibrium. Mm-hmm. And so as you shift concentration, you might for a while have one reaction go a little bit faster than the other for a while. And then we might have metabolism go in the opposite direction. But at the end of the answer for Mark, I was saying that ATP, since it's the fuel for all of the, not all, but many of the productive steps in the cell's function, ATP is constantly being depleted. And so on average, metabolism runs forward.
Yeah, okay. So when we get closest to the state where it's in like total equilibrium, how can what should I be picturing at the macro level? What's my body doing at that point?
When you're not metabolizing anymore?
Yeah.
Well, I would ask, why aren't you metabolizing? Why aren't your cells doing their work? And the cells, the cell is a non-equilibrium system, meaning you have a high concentration of molecules inside the cell that can actually go outside the cell through some of these pores. And so the cell uses the ATP to keep pulling those things back in. And so when your cell stops burning ATP, those molecules just start diffusing away and your cell dies. And so when you reach equilibrium is when you're dead.
Yeah.
Okay, fair enough. So what are the things that can cause this system to go haywire? What are the diseases that cause it to operate less efficiently or less functionally than it should?
So here's where we're going to run into the limit of my knowledge. I'm actually not a physiologist and don't study diseases much, so I don't have a whole list of diseases off the top of my head. We were talking at home the other day about how when this enzyme right here doesn't work that well, then you can get a metabolic disorder.
Okay.
And so you'll end up feeling tired, and most metabolic disorders manifest themselves as just always being tired.
So that's things like diabetes or hypoglycemia, things like that? Those might be mild versions of it, I suppose. They could be pretty severe.
I think that those are actually further up from here. Those are at this step to go from here to here. And all of the regulation that goes from the food you eat to making sugar, there's a ton of biochemistry there that I don't have any slides on. And I would say diabetes is actually a disease of those pathways and not the further down ones that I'm talking about today.
Okay, thanks.
And some of the example enzyme diseases would be some of these congenital disorders that I was just saying, and medically there's sort of some of these rare diseases where there's you have abnormal enzymes, and so the metabolic processes get blocked, and you end up with these what's called storage diseases, or some other. I mean, that's where you're at the enzyme level, where there's imperfect enzymes. It's usually, it usually creates, and they accumulate, and you back up at the point where the enzyme isn't working there. So you get these rare diseases there, but those are kind of uncommon at the enzyme level.
Yeah.
Which I think you were talking about at that level,
And that reinforces this point about the importance of information, because when you have a anomaly, then what that does is it causes the protein that's encoded to have changes in the amino acid sequence. And then that causes a change in the structure of the protein so that the protein doesn't function as well back in these chains. And so it could be as simple as a single nucleotide polymorphism, where you just change one of the nucleotides here, and all of a sudden, boom, the person has cystic fibrosis. There are more complex diseases too. Not all of them are single nucleotide polymorphisms, but it's amazing that so many diseases are actually caused by just single changes in the DNA. That tells you how important this information is, that it's very precisely defined. Okay, any more questions before we talk about evolution? Okay, so, usually everybody's on the same page when we get to this. Everybody's like, wow, yeah, molecular machines are amazing. Isn't it great how that evolved? And that could evolve over so many billions of years. So let's talk about where this information comes in, because everybody's in agreement that the molecular machines are amazing, that they have to be very precisely defined, that there's information there. And so the question is, where does the information come from? And so Darwin's idea is rooted in dog breeding. That's the example he gives in The Origin of Species. And so you can see that by breeding dogs and selecting for certain traits, because so much variation in the dogs, some are small, some are quiet, some are fast. By selecting those animals that have the same traits and breeding them, you can actually emphasize those traits in the gene pool. And by doing that over many, many, many generations, we've been able to make really small dogs, really cute dogs like the little poodle that my mom has. Buddy, this is a shout out to you. Or you could get these really big dogs like this. You can select for certain properties like being able to dig well or be able to chase birds well. So humans are able to design dogs by doing a process of selecting on the natural variation that exists. And so Darwin saw this and he's like, wow, we can design that way. And when he was taking a trip to the Galapagos Islands and he saw a lot of variation in the bird, in the finch beaks, and he saw that some birds were well suited for certain environments, like eating seeds compared to eating insects, and that the number of seeds versus the number of insects kind of determines the size of this population. He had the idea that, well, instead of an intelligent human doing the selection, maybe it's actually nature that is doing the selection on the variation that naturally exists in the population. And so he extrapolated this process to not only being able to produce variations in phenotype like this, but to get very substantial variation such that these birds can no longer mate with each other, and then we'd actually call it an individual species. And so a new species is formed. And building off of Darwin's work, when we learned that the genetic characteristics that are passed on are actually inherited through DNA, and that variation is caused by DNA mutation, that led to what we call the Neo-Darwinian Synthesis, which has been around for many, many decades. And so it's kind of old evolutionary theory right now, and we continue to refine evolutionary theory, trying to figure things out. And this process of variation and selection, we can actually study that, or we can actually use it to solve problems. For example, the traveling salesman problem can be solved using a process of variation and selection. So we would call that an evolutionary search. And we can model evolutionary searches. We can even study it mathematically. And by doing modeling, we can learn its strengths and its weaknesses. And specifically, its strengths and weaknesses with regard to one of the questions that we have right now, which is, can we create new information? And that new information that would be to new species or optimizing proteins and organisms in nature. That's the big question we have right now. Can evolution do this? So let's study how well evolution is able to create new information by doing this modeling of evolutionary processes. And so we start by just talking about search spaces. Let's say we have the search space of ASCII characters. Now, an individual ASCII character is coded by an 8-bit word. That means that there are 256 different possible ASCII characters. And if we were to play the game, pick a letter, I would just start asking, which one of these 256 am I thinking of right now? You guys could start guessing. And you guys could guess and guess and guess. And on average, it would take about 128 guesses for you to guess the one that I have in mind right now. Okay? That is called blind search. We're searching a space that is 256 options large. Okay? Well, can we use an evolution algorithm to get to that guess more easily? Well, to do an evolution algorithm, you need what's called a fitness function. And what that fitness function is when you do a guess, you compare that guess to the fitness function, and the fitness function tells you, is the guess better or not? And the fitness function for blind search, that game we just played, looks like this: completely flat until the answer is right here, and you say, yes, you got it right. Otherwise, completely flat, just no, you're not right, you're not right, you're not right, you're not right. Okay, and if we were to run a search, here I'm plotting time axis as a function of the space that we're looking at, and I just started with a random guess here, and then propagated this with a bunch of variation. And so here we are looking when it's just no, yes. And once we clicked on yes, then it kind of held that one. And then the red curve is actually the variation of guessing after you've already gotten it right and you're kind of staying on the right answer. The algorithm still allows you to try and vary and say, well, does it get better? And the answer is no, there's only one right answer. So it just kind of moves around right here. Okay. I've tried to teach this one many, many times, and it's pretty complicated. So I actually want to pause here and see, are you guys with me on this? My graphs here. Fitness is, I'm asking, is it right or not? And then this time versus the space. Maybe I should move on to the next one. Okay, now there's another game that kids play, not just blind search, but it's like, oh, you're getting warmer, very warm, oh, you're hot. Oh, you're really hot. Oh, no, you're really, really, really hot. Okay, that is a fitness function in search. For example, down here, if we're really far from the target, we would say, oh, you're so cold. Oh, you're in the freezer over here. And then you were to guess something else. And maybe if you were to guess this one now, a little bit of variation, then the answer is, oh, you're warmer. That's better, but you're still quite cold. And then you would guess again. Maybe you might go back here. Oh, no, you went the wrong way. Much colder. And then you'd guess the opposite direction. Oh, you get warmer. And if we were to have a fitness function, which is smooth with a single maximum, you could actually get to the result very quickly. You just climb right up this fitness function to the peak very easily. And what an evolutionary algorithm with a fitness function that looks like this would look like is you might start with a guess that's very cold, but with almost every single increment, you'd actually be increasing in fitness until you get right to the right result. And so this orange curve is the measure of the fitness. And you see that we just climbed right up that fitness function until we're at maximum fitness. And notice that this took less time than if we were to do the blind search. So that means that this fitness function is much better for giving this result than this fitness function, blind search. Now, there's not just these two types of fitness functions. There's another fitness function. What about this one? This one has a global maximum. It also has a number of other maxima, but these are at lower energy. And because it's a local maxima, you actually have to kind of go in the wrong direction in fitness for a while to be able to move away from this local maxima to be able to get away here. And so if you were to find one of these local maxima, it might take you a little bit of variation. You might vary and you're not very far enough. and then you would just be pushed right back to the local maxima again. But if you got a big variation and you jumped out of here, then you might be able to find your way up the next hill. And so that's what this search looks like. Oh, my cursor's moving very slowly. And so, look, you can actually see we started farther away, and we kind of stay in a minimum for a while. I'm sorry, a maxima for a while, but it's only a local maxima. And then there's a big variation, and you're able to jump out of that and find another one that's a little bit more stable, and you stay there for a while. And so, you know what this looks like? This looks like punctuated equilibrium, Stephen Jay Gould's way of explaining the fossil record that we observe in nature. Because in nature, there's actually very, 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 very few transitional forms recorded in the fossil record. Most all the time, we just see the same structure over and over and over again. And it took biologists a long time to admit that. They kept saying, well, we're just going to search hard enough. We're going to search hard enough, and then we'll keep finding transitional forms. And then finally, Stephen Jay Gould just said, we need to face up to the facts. There's very few transitional forms. So we need to change our theory to match the data. And that is that when evolution does happen, it's actually a pretty substantial jump. And we just go from a very stable form to another form that's quite different and also very stable. And so this is what that kind of fitness function might look like to produce punctuated equilibrium. And so we now may ask ourselves, well, which fitness function should there be? And the answer is there are a lot of fitness functions. Because remember, we're trying to search for this single answer. And I've tried to simplify this problem. We're just searching for a single character, B, amongst the whole space of ASCII characters, 1 out of 256 options. And how can we get B? Well, we would have to create a fitness function to have a successful search. And as you look at this fitness function has a lot more information than the target that I'm looking for, because the target that I'm looking for is B. B only has 8 bits of information in it. But I need much, much more information, because for all the other options in the space, I actually need to be able to give you how much fitness there is. And so this actually has a whole space in and of itself of values that it can be. It could be 0, like in blind search, or it could be right here, or it could be a little higher, or there could be a fitness function that kind of looks like this. I drew a bunch of them when I was making these slides, and I thought, I'll take them out because it might get a little bit too complicated. But the number of fitness functions is actually almost infinite compared to our search space. And it's a whole nother dimension of information. So let me say this. There are multiple dimensions. If we do a single letter in one dimension space, that requires a 2-dimensional fitness function, right? The y versus x here is 2 dimensions, when we're only looking for the answer in 1 dimension, x. And if we were to have a sequence of n letters, that creates an n-dimensional space that requires an n+1 dimensional fitness function. And so here I just drew a search for PA, my initials. And so that's now 2 letters, which gives you 2 dimensions here. But we can have all sorts of different shaped fitness functions to be able to try and find the solution PA. Now, I've drawn all these fitness functions, which would lead to successful searches for the thing we're looking for. But there's other fitness functions that would lead to another result. And how do we select amongst fitness functions that are going to give us the result we want versus the one that we don't want? There could be all sorts of fitness functions that lead to a protein sequence that doesn't generate a good protein. I should have said amino acid sequence that doesn't lead to an active enzyme or something like that. How do we do this? Well, what it is that the information that's required to build the fitness function or to select the fitness function from among all the other fitness functions is far greater than the actual information that we're looking for that's contained in the research result. And we were looking at just the information in the search results in the previous section when we were talking about all those biochemical pathways that need to have this enzyme that is structured in just this way. But the fitness function that is able to produce all those enzymes that are organized just right and that are shaped in just the right way with just the right amino acid sequence is much more information. And so we don't actually solve the information problem. Okay, so I put it here. Using an evolutionary search mechanism, Darwinism, does not solve the information problem and the need for intelligent agency to provide that information. You actually need intelligent agency to say, we're going to take this fitness function and run this fitness function with evolutionary search to be able to get a successful result. All right, so that brings me to my concluding slide. And this is why I call this Maxwell's angel. So what we've talked about today is we first learned that from Maxwell's thought experiment, that information is essential to make efficient machines that convert one form of energy to another. And it's very natural for us to seek what's the origin of this information that's used to create these awesome molecular machines that we see in nature, especially since undirected evolutionary mechanisms are impotent. So that's what I was just saying with that last section. As we study evolution and we learn about fitness functions, we learn that evolutionary algorithms do not create information. You actually need more information to be able to get it. And so if we're looking at where the source of information is, it's not evolutionary algorithms. They don't create information. The information is exogenous. It needs to come to the system. And as we think about bringing information theory to thermodynamics, we see that Maxwell's thought experiment shows us that, this section, let me read this one first. Okay, so the Greek word for angel is egelos, which also means messenger or the one who imparts information. And so this should be before this. And so an angel brings information. And so what we see from Maxwell's thought experiment is that we need information to come into the system, to be imposed on the system, to be able to make it into a useful machine that can do work. And so that's why I think it'd be much better that we call the thought experiment Maxwell's angel, because we need that messenger bringing information to be able to make our machines. And I'll be explicit about, what I think is the messenger here. Colossians 1:16, which was written by Paul the Apostle, who I'm named after. He wrote, "For in him," referring to Jesus, "all things were created, things in heaven and on earth, visible and invisible." Okay, thank you. Any more questions?
People are welcome to unmute themselves for questions.
All right, Peter raised his hand. Let's hear it.
So you showed those graphs. I think it was the slide before this, or was it one? Yeah, those. This was really hard to follow. Okay,
that's what everyone says.
Yeah, because you've got four things going on in each one at the same time. But there's something that I think you did not say. I didn't hear you say it, which is that the path depends on the fitness function in some way. So I didn't hear you say, but I think your assumption is that the path that you take here is dependent on some randomness plus the fitness function, right? Like it's
yes. So evolution is variation and selection. So the variation is the randomness, and the selection is when you compare against the fitness function, or when you impose the information of the fitness function on the search. And so the trajectory that you take, or the path that you called it, is determined by both the variation and the fitness function.
Right. And I guess there's some little formula that you could have written there, but maybe that would have been too complicated to write. I'm not sure how this is done. If you've got some really complicated idea of like trying to analogize genes with alleles and stuff like that, or if it's simpler than that.
This is definitely a model. that is used to mimic it. And so what I'm doing is I'm saying I have some starting position, and from that starting position, I add some variation. So I move from here in some random direction. And then in my new position, I ask myself, based on the fitness function, is my new position a higher fitness or a lower fitness than my previous place? If it's a higher fitness, then I keep that.
Oh, okay. Well,
if it's a lower fitness, then I throw it away.
Okay.
Now, to be able to get so much noise here on the search, I included in my selection step that even if you take a backward step, as long as it's not too far backwards, we'll still keep it. Because if you only select the ones that are absolutely progress, then these searches are very fast. You just run straight up this. But if you have that kind of search where you only accept things that are better, you will never, ever get out of a local maximum too.
Right. Yeah.
So to be able to use the same algorithm for all three of these searches here, I included during my selection step some tolerance for going backwards.
So Paul, while we're on the topic of fitness function, I'm trying to understand it as well. Does the fitness function in effect provide some feedback to the searcher then about the quality of? Okay.
Yes, exactly. The fitness function is in the game that we were playing with warm, cold. The fitness function is the person that has the knowledge of where the target is.
Right.
Telling the searcher, oh, you're warmer. Oh, you're really hot. Oh, no, you just took a cold step.
Okay.
Okay, that's the fitness function, is that communication of you're getting better or not.
Okay, good. I just wanted to, thanks.
Well, I don't know if Bob still had a question, but Charles has now also raised his hand.
Yes, I've heard, I'm unmuted, yes. I was introduced by a math professor years ago to the idea that there simply wasn't enough time since the creation of the universe for random mutations to produce DNA, which I think is true. My question is more, if it is the logos, which is conforming evolution in a certain direction through natural laws, what is the magnitude of the complexity of the logos needed in order to produce DNA? I mean, I don't even know how to approach that as a mathematician, but it seems to me that you need something very, very complex in order to guide a process that would produce DNA.
Yes. Can I take you away from DNA specifically and say an evolutionary search that would optimize an organism that already has DNA and is already able to function in terms of being able to reproduce and is already functioning in terms of having some variation due to changes in the DNA. The whole problem of how do you get the first cell and the origin of life it's extremely difficult, and it completely defies evolutionary algorithms in my opinion. And that's a book written by Stephen Meyer called Signature in the Cell. I highly recommend it. But I say that's a separate problem from evolution, because evolution assumes that you have an organism already. But what I'm trying to say here is that to do a successful optimization using evolution, you need a lot of information, more information than what's contained in all the DNA. Like the DNA codes for the amino acid sequences, and the amino acid sequences code for the fold of the protein. The fold of the protein codes for where things are and if they're at the right distance to be able to do their job. And there's so many of these proteins in our body. There are thousands and thousands of genes. encoding for thousands and thousands of proteins that do their specific task. So much information. And whatever is guiding that process has more information than that, right? Because that search for that information is like just this single result that I'm trying to depict here, finding this single point, B. But to be able to successfully do that, you need to know if you're fit when you're far from B also, and how fit you are far from B. And it's an incredibly large amount of information more. It's a whole nother dimension. And so, what is that logos? I mean, that's why so many of us are so quick to attribute that intelligent agent to somebody who also we would call God, all powerful, all knowing, and stuff like that.
Okay.
Thank you.
Can I ask a question?
Yeah.
I'm not sure what raising a hand. Is it literally raising a hand, or is this an electronic hand I'm supposed to click? I don't know.
There's both.
I haven't discovered the electronic hand. I have two questions about the search. The first one is that those little local maxima, you have multiple maxima, there's no guarantee that those multiple maxima will be small in range or in height. Some of them could be quite wide, which would make it more difficult. And then the second question is, is it reasonable to assume that there's just a single target? There's only one target that we can anticipate as humans, but there may be other target possibilities that we haven't considered that evolution might conceivably have directed towards. And if there are many of those, it might make the probability of achieving them that much easier. Is that clear what I'm saying? I'm not sure.
Yes, but there's many different aspects to what you're saying, because we could talk about organisms, individual organisms, in which case, and I said this search right here on the right mimics punctuated equilibrium, where you might have one organism that stays for a long time and then it changes a lot and you get another organism. And what we see in the fossil record is that the number of organisms keeps growing. Some go extinct and some and we get the proliferation of new species. And so multiple species can exist side by side.
Mm-hmm.
I would say that's actually part of the beauty and complexity of the fitness function that led to the world that we see today. There could be a fitness function which would say life must be only this one thing, and it would absolutely wipe out everything else. But that's not what we see. We actually see that life lives in ecosystems that are so beautifully balanced, with each organism playing its part to support each other. And it's very symbiotic. And, if we're philosophizing and extrapolating, we might say that symbiosis actually reflects the character of the creator, who actually, lives in three persons together.
Thank you.
There are some hands raised, but I can't tell if they're new or old. So if you have a question, just unmute yourself and please ask it.
Well, I've just been having a problem with this whole idea of a fitness function. I mean, first of all, the universe wasn't taught, so I'm not quite clear. The only thing I can see is either, at least evolutionary, is how many descendants are you able to produce and how long are they able to live for? It's kind of, and it's kind of like, it's not like there's a perfect answer necessarily, and it's just you do better or you do worse kind of thing there. But I don't know what kind of fitness function is when you describe me as some kind of feedback. You're doing a strategy and the fitness function is telling you how well you're doing there. And the universe seems it's either either your descendants are going to thrive or they're not. And the other day, of course, and then the goalpost keeps moving because environments change over time. So I'm not really quite sure what this is.
But Bob, like
I'm
being sterile or starving to death, that's a pretty strong feedback. To be sterile or to not be able to feed your progeny, that's pretty strong feedback right there. So
right,
I would say that the nature can provide this feedback.
Right, but all these things take generations of time, but I'm not quite sure how you go to it. It's moving to. I mean, I'm just finding it hard to see how the randomness is going to what it's going to produce, because not only you're moving towards a local maximum, but the environment changes. So what works today may not work tomorrow, and you're probably never going to find the perfect answer. I mean, I think you're never going to find. There is no perfect answer you're going to find. So it's kind of like. It's a very fuzzy. It seems like it's a very fuzzy situation here.
I agree that this is very complex.
Fuzzy.
What is reality?
Fuzzy,
is very complex, and here this is the bare bones, minimal simplification. And so this does not capture reality, but it's trying to give us a hint, and it's trying to teach us the principle that. If evolution was the way by which we got speciation, then that process would require more information than just the result. My only point here is to say evolution does not create information.
That was the point, I think, of when we did that signature in the cell. Was it that we did that some years ago? I think that was kind of Meyer's point. Was you can't create meaningful information out of nothing.
Out of nothing.
That was what his conclusion seemed to be, if I recall. That's what he was going to make the argument. You had to have.
And so
the point of this source providing the information that you were working from.
Yeah, I mean, the point of this argument is actually not to describe how evolution happened.
Mm-hmm.
The point of this is to say that there's two sides of this camp. There's people who say that there's philosophical naturalists that say all you need is energy and matter. And if you just have energy and matter and you do evolution, you can get information. And I'm saying that is absolutely wrong. Everything we understand about evolution actually says you cannot create information. And so then you need something else. You need a system of thinking that allows for information to be put onto the physical world. And that's why I started with that C.S. Lewis quote, because there's the physical world, there's the laws of physics, the laws of nature. And they say, when you do this, and then you get this, and then you get this. And what intelligent agents say is they say, oh, don't start with this, start with this instead. And so then the laws of physics say, okay, I'll start with that then, and I'll do this and this and this. And a miracle is not a violation of the laws of physics. A miracle is just imparting information to the physical world from which the laws of physics then proceed. And I would say creation, the ability to make these amazing molecular machines is imparting information. They're miracles.
Let me ask along those lines again, though. When you said that evolution cannot create information, in an absolute sense, you're saying that the whole universe, the information content is constant? Is that premise?
No, I'm saying that the universe is open and that information is put into the universe.
Okay, but even if the information content of the whole universe was constant, using the thermodynamics and entropy and all that, isn't it possible that with energy that you have localized increase in information even though some other parts of it may be losing information, some smaller part of it may be increasing information, right? I mean, that's how the whole, the sun coalescing out of a cloud and then so on and so on. Even though the universe as a whole, the entropy is increasing, locally in our solar system, entropy has decreased, and that's why life can exist on Earth. So I guess I'm wondering, the fitness function not being able to be more complex than the end result without extra information, it doesn't seem like, couldn't the information be there except that it's not uniformly distributed?
So I would like us to be careful here. From the things you're saying, I can tell that you've read a little bit more widely about entropy and information, and I didn't actually touch on this, but it's very common in the physics literature to equate entropy and information. And so we talk about like Shannon information, which is just a measure of the entropy. And it goes back to, this whole thing about the number of states gives you the entropy. And so as you have more and more states, you have more entropy, and we can measure this in bits or in information. I am not using information in the same way, because when those physicists use information, they are just simply saying, how many possible states are there? And I'm using information differently. I'm saying that I want, out of this whole selection of all the different possible sequences of ones and zeros that could be, I'm wanting to select this one sequence that opens the door at just the right time so that you get productive work. And so this is a very strong narrowing or whittling down of the entropy. And you can't get this simply by passing energy through the system, like you can when in a just natural system where you pass energy through a system, an example might be a polar vortex, which, the energy is there because of the imbalance of the heating of the poles versus the equator and the whole convection of the atmosphere. And at times you get this random fluctuation that pushes really cold air down and we get hail. Okay, so yeah, that hail is a low entropy system. But that happened because there's a lot of energy that was put into the system by the sun. I'm saying we need to actually select some specific states, and that's the information. We need to organize matter in very specific ways to be able to then get useful work. And one of the ways I've been trying to think about this to differentiate it from natural fluctuations in local entropy levels due to heat or energy throughput of the system is to talk on the scale of dissipation or how fast energy is able to be transported in a system. Our machines, they work on a scale of energy transport that is much smaller than what you find in nature and the variations in energy that you find in nature. We organize matter to purposefully separate energy into very small compartments, like a cylinder, which is much hotter than the block of the engine. As a matter of fact, we purposefully flow coolant through the block to keep the block very cold, and we separate that energy at a very small length scale that never happens naturally due to natural dissipation of heat or natural convection of heat, diffusion or convection. Does that help, Young?
I'm still not understanding, I guess. I mean, in terms of the energy flow through the system to decrease entropy, I mean, clearly you gave an example like of air conditioners where you supply energy in order to get low entropy and so on. But the question I had was, why is it that you think you need to a priori specify the fitness function? Because that seems to be the crux of your argument, is that because the fitness function is more complicated, mainly requires more information, that evolution cannot explain the complexity. But why is it that a particular fitness function has to exist in the first place? Because it seems like the point of the mechanism, I mean, not necessarily that I agree with it, but the point of the Darwinian, the survival of the fittest, which is kind of a tautology, but still, it's just saying that things will happen and some things will result and other things will not. And so it doesn't seem like there is a priori a fitness function that is required.
Okay. I'm actually not trying to say that from the beginning of time, there was a single fitness function. I'm trying to use evolution and our understanding of evolution to defeat philosophical naturalism. That's my only goal here. As a matter of fact, I don't believe, I'm not a theistic evolutionist. I think there's too many problems with the actual fossil record to think that the Darwinistic mechanism of smooth transitions was what happened in reality. So let me state again, what I'm trying to do here is say that even if we thought within the evolutionary framework, what would be required for evolution to be successful is more information. And where does that information come from? Information is exogenous. It needs to be imposed on nature. And it comes from the intelligent agent, the creator, God. And then how God does it. Now that's a whole nother set of questions. And I don't have time to go through all of that. It was brought up. How did the first cell form? I mean, that's a really big one, really hard. But once you have that, then, what do you do? I mean, clearly, I think we see an increase in complexity over time, but we also see a lot of consistent, a lot of homeostasis, where the same organism stays there for a long time. And so my point is not that there was some big fitness function that was boom there at the Big Bang that has been guiding and directing everything from the beginning of time. I would say that's actually a deistic argument, and I'm not a deist, I'm a theist. I think that God has been working throughout history. I think that God created nature with the characteristic of being open and needing, maybe not needing, being open and being receptive. As Lewis put, nature is a something hostess.
Paul, wouldn't it also be worth saying that the Anthropic Principle itself is evidence of the exogenous nature of information crossing the threshold into this universe? In other words, yes, the universe is an anthropic system that is winding down, but the laws of physics themselves do not change. Something holds them fixed. So,
I mean, I think that's true.
Right.
But I think that's also a separate argument.
Right, that's a separate argument. I'm just saying that if the universe is not a closed system, which is what naturalists believe, then information has to be sort of a constant raining, you might say, across this threshold to make human life, planets, and many other things operational because you can't explain it in terms of simply the probabilities. And that one slide you had about the probabilities, you might want to just point out the discrepancy, even to create a simple
protein.
A simple protein, right?
Yeah.
And so if you believe that the universe was infinite in age,
the universe is finite.
Right.
And I think it takes a whole lot more probability resources than what is available in the universe to be able to get the information that we have in just one of those proteins if we were to do it by an unguided search. Yeah.
Can you quickly show that one slide with the probability numbers again?
I gave here, if we have the 34 characters. So we're just looking for a sequence of information. And here I did that Colossians 1:16. For in him all things were created. This is 34 characters. If this is all the information we're looking for out of an evolutionary search, that would be 8 times 34 bits, which is 272 bits of information, which is a total of 10 to the 82 possible sequences. That's a lot. That's a big number. And there are 10 to the 80 particles in the universe and only 10 to the 18 seconds. So this kind of gives us our probability resources. Now, if we were to do a search for this sequence where we had every single particle in the universe, every single second, give us a random sequence. Well, yeah, for the age of the universe, we would have stumbled upon this. But all I have to do is just double this in length. And then that makes this 10 to the 164. And now 10 to the 80 particles times 10 to the 18 seconds. And since Planck time, I think it's 10 to the minus 34, we could say that's 10 to the 52. And so that would be 10 to the 132. 10 to the 132 is still less than 10 to the 164. And so all we need to do is just have twice as many characters of this, and the probability resource of the whole universe is not capable of generating this random sequence. I'm sorry, this specified sequence by selecting random sequences, choosing from random sequences.
And of course, this is nothing compared to creating an amino acid.
Compared to creating a protein, right?
Or a protein, right.
Yeah. So I mean, these numbers blow up so quickly, and it's sometimes hard to wrap your head around how big the numbers and how quick they are to blow up. And maybe I should have put a slide in to try and really watch that blow up. But I mean, that was actually kind of my point in just going from this, which is a single character, 8 bits, really easy to guess, to 2 right here. And you need a more complex fitness function. But that's just 2 letters. I mean, it's already, it grows exponentially, right? Very, very fast growth in the search space. I really appreciate you guys all being here. Thank you very much.